Okay. I never know quite how to present how detailed is this, but you know, obviously, as I said, cyanobacteria are around a long time. People have studied them for a long time. But in terms of the harmful algae bloom situation, it was really, well, you can go back as far as the 60s and actually started here in California with red tides and shellfish poisoning with the paralytic shellfish poisoning with the saxitoxins. So actually California was, you know, sort of where it started. There was a fellow named Ed Shantz who was out here, I think he was UC Caltech, and he actually did the chemistry and biochemistry of saxitoxin, the very first identified red tide toxin through shellfish vectoring. And he eventually went to work for the Army at Fort Detrick, hence that's why saxitoxin gets in with the biological warfare materials, unfortunately. And much of the work was done at Fort Detrick with regards to the toxicology and production. Fort Detrick still has probably several grams of pure saxitoxin hidden away somewhere there. Anyway, so let's just kind of back up and talk about cyanobacteria. I do point out that, again, there are evidences that it was recognized for a long time, including Will Shakespeare, who said, "There are sorts of men whose visages do cream and mantle like a standing pond," and we all accept that that's going to be a Luger Bloom, right? The Merchant of Venice. But there's more than that. We have, you know, anecdotal things in the literature about poisonings and colorations of water. Even in China, when I was working there, there's an ancient military handbook for generals, and they actually refer to wading across rivers and having the soldiers drink the green water and then dying on the other side. So, and that's, you know, that can't be anecdotal because all of that's got to be real, right? But the factors in the formation of cyanobacteria: urban agricultural and industrial expansion, increased nutrient input, especially nitrogen and phosphorus, and I like to emphasize both. We used to kind of think if we controlled just phosphorus, we were all set. Really, you got to focus on both. You can't do one without the other. Water use and hydrologic changes, diminished water flow and less mixing, and of course, during a drought, that's definitely what happens. There's hardly any water going in or out. And climate. Climate does play an interactive role. It doesn't cause blooms, but it certainly allows blooms to come on earlier, stay on later, hence the problems and exposure for health risks are extended significantly to some situations now where these blooms are with us year-round. And of course, that didn't used to be the case. So, as a consequence, blooms are more common, more intense, and of longer duration. And that's what brings us kind of to the current situation. Okay, this little cartoon then tends to summarize what the issues are, and I think everyone recognizes this. You don't always have everything together, but this is what it takes to kind of give you a good bloom. We're obviously having developmental issues with nitrogen, phosphorus flowing into the systems from agriculture and forestry, and you know, management around lakes and reservoirs. You even have air pollution putting nitrogen into the system as depositional materials. Then we've got, you know, our conditions in the environment that create turbulence or lack of turbulence or stratification or lack of stratification. Cyanobacteria are lazy. They love to grow in quiet, non-stratified, non-mixed situations. And of course, all the reservoirs we've built since the Second World War kind of qualify for that in most cases. So that's why we're getting to the situation now where those reservoirs are supporting them. Rivers are often flowing and mixing less than they used to be because of whatever situation. So this kind of puts it all together. A recognition really in the literature, at least the formal literature, in this case the scientific publication Nature, in 1878 there was an Anglican minister in Australia named George Francis who did more than give sermons, I guess, because he was watching the lake. And in Adelaide, which is in South Australia, there's a brackish water lake, which they've now dammed, and it's mostly fresh, but it's almost an estuary thing. Noticed a sheep poisonings from these blooms, and he published this short note in Nature in May of 1878. So that's kind of where we document the original first reported cases of animal poisonings from cyanobacteria, drinking cyanobacteria blooms. But if you go into the non-formal scientific literature in the U.S., i.e. newspaper articles, there's quite a number from the late 1800s, early 1900s, about mass mortalities, especially in the Midwest, Iowa in this case. And these involve situations where the blooms come late in the year, August, September, even into October, and migrating birds that are going back and forth. You'll get 5,000 birds land and they're all dead the next morning. Or a farmer sends his cattle down to drink in the morning and he gets 50 dead cows. These are the kinds of mass events that sort of triggered farmers, at least, to pay attention to their water supplies as early as the late 1800s. And we've had documentations like that ever since. So natural eutrophication, in other words, lakes that age and have normal nutrient levels that are high enough to support cyanobacteria, have always been an issue, and especially in those cases where the lakes are rather shallow and maybe big and broad and are nice cultured tanks for the algae. So we recognize this, but what has triggered the changes is everything we're doing to lakes and reservoirs in particular that allow us now to see more of these events. So, but we can go back that far and feel comfortable that we've had the. So I guess the next official sort of recognition of this issue would have been the 1970s, early 1970s, when really the environmental movement took off with regards to nutrient pollution. And that was Lake Erie in the 1970s, when the acronym was Lake Erie was dead. Well, Lake Erie wasn't dead, but it didn't look very good. This I got out of the archives at Ohio State University, and pretty well shows you what we had at that time. Now, nobody really recognized that there were even toxins in these organisms at that time, let alone what the issue might be. It turns out that this is probably a Phanozomenon flossaqua, a nitrogen fixing. So even back then with these blooms, we had a nitrogen limitation, and it was probably primarily phosphorus that was driving this system. Now we have so much nitrogen and phosphorus that we're getting the non-nitrogen fixers, which are more commonly toxin producers like Microcystis, which is what we'll be looking at today, and hence the shift to that. So as you can see, it's been a progression of events and situations and circumstances that sort of bring us to this. This particular species, Anabaena Phanozomenon flossaqua, is not a toxin producer. This is the same one that's harvested up at Klamath Lake for health food, by the way, those of you that are up that way. It's generally recognized now, though, that all states have some sort of issue and reporting process with cyanobacteria. So cyanotoxin poisonings and health issues in red, cyanotoxin poisonings and health advisories even if you don't have documented poisonings. So it's ubiquitous. It just depends on how proactive that state has been in monitoring and setting up and sort of documenting what really happens. Okay, this is what we're often met with then in a sort of normal native population of cyanobacteria that has a mixed population. And this actually shows you most of the ones that we want to become at least passingly familiar with after the end of the day. Almost all of these are capable of producing toxin. That doesn't mean they are producing toxin. So when it comes to management and risk, you have to decide, am I going to just identify and post? Am I going to identify and count and post? Am I going to identify, count, and do toxin testing and then post? Those are the kinds of issues that you have to decide when you set up your monitoring program. But hopefully by the end of the day, if you don't already know, this is going to be Microcystis. In this case, Microcystis aeruginosa. Each one of these little dots is a cell. This colony, which is an accumulation of cells, is a couple hundred microns across. The straight-chained filamentous ones, we look for little heterocysts in here, so we know that it's a nitrogen-fixing cyanobacteria. This is most likely an Anabaena, or maybe a nostoc, most likely an Anabaena. This is certainly an Anabaena, probably Phlos aqua, one that is a colony, and the filaments all twine around the cells. So that defines a colony. Accumulation of cells or accumulation of filaments defines a colony. And that's a key way that we go about identifying these groups. Here's another colonial one related to Microcystis called Cielocerium. And here's a Phanosoma on Phlos aqua, an entire colony of one. So those are some of the key ones that are going to be producing toxins. So we're not talking directly about toxins so much, but I did want to point out the different toxin-producing groups of algae, in which case this includes marine and freshwater. So types of harmful algal blooms. By the way, harmful algal blooms is not a new term, or is a new term, I should say. When we started making our case to Congress in the early 80s, and we had one of the first congressional acts that supported the study of harmful algae from congressional monies, we had our series of meetings, and one of the key people working with the red tides, Don Anderson at Woods Hole, promoted this term harmful algae that would be recognizable and we could use in white papers and sort of thing. So it's really since the 80s that we've sort of used this term HAB, or harmful algal bloom, to define algal blooms that are harmful. And it doesn't mean that they're all toxic. They could simply be producing very dense blooms that lead to organism stress and low oxygens and things like that in water supplies. Or they could be the forms that produce potent toxins that lead to illness and death, either directly by consumption of the water containing that algae, or the toxins mean the algae being consumed by organisms that concentrate the toxin, and they go up through the food chain, and then even people will consume them and cause illness and death. So most of these are marine. So these dinoflagellates would be all marine organisms, which of course is what you would have issues out here on the coast, especially paralytic shellfish poisoning. Some of the newer ones, this one's been known since the 60s, but all these others came along more in the 80s in terms of recognizing different kinds of harmful algae poisonings. So we have acronyms like diuretic shellfish poisoning, neurotoxic shellfish poisoning, ciguatera fish food, estuary-associated syndrome, and even ASP. The ASP is kind of particularly important here because you have a massive bloom, especially in Monterey Bay and up the north coast areas of this organism, which is a diatom, not a dinoflagellate, and produces this toxin leading to amnesic shellfish poisoning, which is called domoic acid. And I'll touch on this in the lecture on taxonomy of HABs in a little bit. And then finally, cyanobacteria toxin poisoning, which we're going to focus on. So quickly, the kinds of bioactive compounds cyanobacteria can produce, they are prolifically producers of bioactive compounds, and not just potent toxins that cause illness or death. They are a major source of pharmaceuticals, antifungal, anti-algal, anti-cancer compounds, and that's a whole different story in itself. That's why I have this grouping called cytotoxins, and then toxins with acute and acute lethal or chronic. So again, we're going to focus on these, fortunately, because otherwise there are literally hundreds of compounds that are bioactive. So some of those cytotoxic pharmacologicals could be antiviral, antibacterial, antifungal, anti-helminthic or against worms, anti-tumor, cardioactive, anti-inflammatory. In fact, a number of these are in commercial production. So and very important. But the cyanotoxins, the ones that I'm focusing on as acute and acute lethal, we're grouping roughly as neurotoxins, liver toxins, hepatotoxins, and dermatoxins, contact irritants, cause you rashes and things like that. These are largely freshwater, this one's largely marine, but all cyanobacteria. How do we become exposed? Well, the obvious: surface drinking water, recreational waters, through the food web, maybe through dietary supplements. This is in here because out of Klamath Lake there's thousands of pounds of algae that are sold for health food. And as I indicated, it's not the Phanozomenon philosopha that's harvested that's the problem, it's the other algae that are contaminated with it at certain times of the year. So that has become an issue. And then this is one of the exposure routes that I investigated using a public water supply that's not properly treated, and the toxins go in and are used in a medical facility, in this case a dialysis facility, because in dialysis you get dialyzed against about 120 liters of water for each treatment, and if that water contained toxin, then you're dead. And that's what happened in Brazil in 1996, and 130 people died. So these are the situations for exposure. And of course, in terms of the other organisms that might be affected out of the water environment, almost anything. We've documented losses from all different groups of wildlife, whether they're birds or fish or wild invertebrates, shellfish, shrimp, that sort of thing. Aquaculture fish. Aquaculture fish and invertebrates are particularly an issue because they're confined in a space, and being confined in a space, they're sort of exposed when the algae are there, and the algae are there because they're feeding these things commercially feeds, which essentially neutralifies that water and causes the blooms to grow even more. So a good example is net pen salmon up off the Washington and British Columbia coasts have a disease called net pen liver disease, where microsystems are produced on the materials that grow on the nets, and the fish feed on that material and have the same liver disease that humans would have. Water users, domestic and wild, animals, humans, and plants. Agriculture plants are an interesting situation here in California, because I'm sure that you are using reservoir waters that have blooms, and you're irrigating those crops with that contaminated water. And interestingly enough, because the mechanism of action, at least for the microcystin, which is the main toxin that you have an issue with, that toxin will actually inhibit photosynthesis. So it's not so much that people are eating contaminated leafy vegetables, but in fact those leafy vegetables are being impacted, and you're getting poor growth and chlorosis from the toxins themselves. So these are the two, virtually anything you can think of that you use water for can be impacted given the correct circumstances. Quickly, the cyanotoxin producers, and we're going to come back to this throughout the day because we need to. And again, this isn't a chemistry class, but I am going to just throw these up so you kind of see some chemistry. The alkaloid toxins, alkaloids are things that have a nitrogen ring kind of compound. The main one you would be familiar with, not personally, is cocaine. That's an alkaloid. There are literally thousands of compounds that are nitrogen-containing compounds that have these qualities, and they're often bioactive. But Anatoxin A is one of those alkaloids. It's the one I did my thesis on, and it is one of the key neurotoxic poisoning compounds that animals are exposed to here in the U.S., including here in California. Anatoxin AS is interesting. It is an organophosphate, just like any of your other organophosphate pesticides, insecticides, has the same mechanism of action and would be indistinguishable. And obviously in lethal doses gives you the same symptoms as a pesticide poisoning cholinesterase inhibitor. The other alkaloid, Cylindrosperm opsin, I think is becoming more of an issue. I'd like to see more work done on this in the West Coast areas and California. We know that toxin's been identified in a few cases in California and Oregon, and I'd like to get someone working on what is the producer organism, because I think it's different than the other ones we've recognized to date. So, and then of course we have those paralytic shellfish toxins, the saxitoxins, which we recognized originally from the red tide dinoflagellates, shellfish poisoning. As it turns out, there are more genera and species of cyanobacteria that produce saxitoxins than there are dinoflagellates in red tides that produce saxitoxins. It's just in the freshwater environment we don't have all of those vectors concentrating the toxin, shellfish in particular, so we don't recognize the issue. And then there's some alkaloid contact toxins, which were primarily found in marine cyanobacteria, but we're now identifying them in freshwater mat-forming cyanobacteria too, here in the U.S. The ones you're more familiar with, if you're familiar with cyanobacteria and their toxins, would be the cyclic peptide microsystems, because, as we know, Microcystis, this non-nitrogen fixer, is becoming dominant in so many of our water supplies. So this is what we have. I have samples of this to look at today that was brought with us to Santa Cruz from one of the lakes near Oakland. And this would be the one in Pinto Lake. If you heard about the sea otter poisonings, that's Microsystem and Microcystis. And related, these are heptapeptides, 7-membered ring, and this is a pentapeptide, a 5-membered ring with similar mechanism, which we'll touch on briefly. And I also think this one should be looked at more in California, because you do have brackish waters which could support the nodularia. All right, so a ranking then of these different toxins. Certainly Microcystis is the one that we'll have most issues with. And then the anatoxins, then the Cylindrosperm opsin, then the Lingbia contact toxins. Maybe, we now know nodularians are in the U.S., been identified in Great Salt Lake and some of those brackish water lakes. But not a lot of animal poisonings documented so far. And then saxitoxins. And then one that's kind of controversial, but if it is true, is significant health risk. It's something called BMAA, an amino acid that is a neurotoxin. And if it's true, it causes symptoms similar to ALS or Lou Gehrig's disease. So, lots of ones to worry about, but I really think that microsystems and anatoxins and these top 3 are the ones that I'm sure you've got in California. All right, the chemistry, we're quick because we don't need to pay a lot of attention, but to show you what I was referring to. Anatoxin A, the alkaloids, these nitrogen-containing, usually ring structures. Cocaine is a 6-membered ring structure, looks very much like that. In fact, the synthetic route to the production of anatoxin used L-cocaine originally. That's a trivia question that comes at the end. Anatoxin AS, that organophosphate, here's the phosphorus. Exact same mechanism as your organophosphate pesticides. And then the whole groups of saxitoxins, these are guanidine rings, and they have all of these compounds. All you've got to do is change, remove a hydrogen and put an OH group, or take out a hydrogen and put in a double bond, and you've got a different compound, so they're called analogs. And so there's probably 15 or 20 analogs of saxitoxin, all of varying toxicity. And then likewise with microsystems, which are these 7-membered amino acids. So we put 7 amino acids in a ring, and we do several different things to them. We have about 125 different analogs of microsystems right now, which makes it difficult because you take an ELISA kit. If you're doing ELISA for water supplies, you don't know that you're detecting all those. They've never been tested. But you're getting 95%. So here we get all of our analogs are our different microsystems because we put different amino acids here, and we put in different subgroups here and there, and almost anything you can think of that makes chemistry sense would give you a different microsystem. So that's why we use these terms microsystem LR. If you've, how many have heard of that particular one? That's the main one. So that would have an arginine and a leucine here, hence microsystem LR. And if it was microsystem LA, which is another fairly common one, we would have an alanine and a leucine here. So that's why you see these. So if you buy these kits and it says this is calibrated against microsystem LR, that's why, because that was used as a calibrator in the kit. It doesn't mean that those antibodies in the ELISA kits don't cross-react with other microsystems, but you're never sure if they're going to cross-react with all 125 because they've never been tested. But they do cross-react with the main ones that are going to be at issue. And then the nodularin, which is the 5-membered ring, looks very much the same, but there are very few, there's only 3 or 4 analogs for that. The real active part of this molecule, if you're interested, is this right here. This is a hydrophobic ring, hence that allows it to cross membranes, and when animals or you ingest the water and the toxins in your gut, it goes through into the bloodstream and then passes into the liver. And the liver is particularly sensitive to it because it passes into those hepatocytes and blocks a key cellular function called protein phosphatase activity. And when you interrupt the activity, you interrupt everything cells do because they are the energy compounds for the cell. And in the liver, if it's there, the liver starts to react and the cells start to swell, and then blood pools in the liver, and then you die from hemorrhagic shock or liver failure. And that's how those 130 people died in Brazil. Okay, Celinda spermopsin, another toxin that you should be paying attention to in California, looks like this. It's again a kind of a guanidine compound, and it has multiple effects. It has organ, kidney effects, liver effects. It seems to be a much more wide, broad spectrum sort of toxicology for organ effect than microsystems. And a lot needs to be known about that toxin as well. The BMAA, the new one on the new kid on the block, is just a simple amino acid, but apparently is responsible for neurodegenerative disease, very similar to ALS or Lou Gehrig's disease. The idea that it might be produced by all groups of cyanobacteria hasn't been supported in recent. There are problems with identifying that compound against all the other amino acids that would be in this organism. So I don't think this is an issue. Don't go run into your supervisor and say, we've got to pay attention to this. We need to pay attention to microsystems, anatoxins, and Celinda spermopsin. Okay, what kinds of legislation have we seen with cyanobacteria at the federal level? Well, within the last month, we've seen more than we've seen in the last 20 years. But in 1996, the Safe Drinking Water Act amendments did authorize regulation of contaminants and establish procedure for making regulatory decisions and allowed the adding of contaminants about every 5 years. Algae, interestingly enough, were put on this list without defining what ones they were concerned about or what toxins they might be concerned about, but they've been there at least on the list. There's no regulations. No one in the U.S., well, that's not true. Oregon actually regulates microcystin in health food products, but not in drinking water. Brazil is the only country that actually has mandatory regulations for microsystems and water supplies. All utilities must monitor and must adhere to this 1 microgram per liter or part per billion as the action level for microsystems in finished drinking water. And that is a consequence directly of 130 people dying in Brazil. Like I said, we're always driven by events. We don't do a whole lot of proactive work. That CCL list is revised. 1998 did include algae and toxins in general. Same thing in 2004. And just this year, 10 years later, 11 years later, there now are some guideline values for microsystems and Celinda Sperm Opsin, just put out by the US EPA in June. Maybe it was May. And we'll cover that in a little bit. But just before I retired in 2006, we sat down with the US EPA and set a priority listing for cyanotoxins. So just like I've said, even here in California, Microsystems, Celinda Sperm Opsin, and Anatoxin are the primary ones. And secondary ones would be your saxitoxins and Modularians. And that hasn't changed, and that's the same what the EPA recommends now. Prior to this time, before the U.S. really got involved, Australia set most of the background materials and guidance that the U.S. and the rest of the world really employs. And they did this because, maybe like California, they've got real water issues. And they build big reservoirs, and they have these periods where it rains phenomenally for a year and then not again for 8 years or 9 years. So they have these giant reservoirs that hold all this water, and of course in those off years, those reservoirs become real culture facility for cyanobacteria. So they've had to pay attention to their cyanobacteria issues for quite some time. Notice all the gum trees, the eucalyptus. So some of those Australian publications that we've used, especially when we did the World Health Organization guideline and states sort of look at, all came pretty much from Australia, including these. Their guidelines in 1996, 2001, 2004, 2004. So those are really what you know, if you were interested, you would go back and you'd look at these, because you'll see that very much what we accept and adopt comes from these documentations. And then the one that gets cited a lot as well, in 1998 and then published in 1999, World Health Organization put a group of us together, about 30 of us. And if you remember back then, it wasn't long after the East Germany became Germany. So to spend WHO monies in East Germany, we had to go to East Germany to formulate these guidelines. So we had a great time in East Germany for a couple of summers. And out of that came this WHO book, Toxic Cyanobacteria in Water Guide to the Public Health Consequences. And I have a copy here if you haven't seen it. You can still get this. It's, I think there's still copies available, like on Amazon. A second volume or a second edition of this is in the works, but something happened with the funding, so it's sitting somewhere as manuscripts. So I don't know if it will get published or not. But I think what's replacing this is, you know, different countries, even states, are adopting and doing their own kinds of documentation. A couple of others, there's something called CyanoNet, which is supported by UNESCO, International Hydrology Program, and we put out a global network for cyanobacterial blooms and toxin risk management, which is available free as a download. It kind of gives the international perspective. We had someone for each continent do a review, and I did the one for North America. The U.S. National Plan for CyanoHABs kinds of struggles along, but in 2008, the U.S. EPA supported, actually the the symposium was in 2005, but the publication was in 2008, and this is this 1,000-page book that has everything you're supposed to want to know. And it's all free from download too. There has been attempts at freshwater harmful algal bloom legislation. Eventually it got put into the marine legislation, which stalled in the, well, it was passed in the House and stalled in the Senate, and just got passed this year, finally. So the new support for the so-called HABs, if you see that acronym, means, you know, since you're all in agencies, you all like acronyms, but it's called the Harmful Algal Bloom and Hypoxia Research and Control Act, or HABs. And that's kind of where it sets for the legislative purposes in the U.S. Other citations, this is our publication on the human fatalities in Brazil. There's some other HAB documents that are focusing on freshwater that are kind of out of date now, 2008. In 1995, we did an American Waterworks Association report that utilities got if you were members of AWWA. Did any of the utility people here, do they have this report? Maybe? If you're a member of AWWA, you got them. And then that proceedings for the US EPA and the Freshwater Control Act as well. Now, bringing us totally up to the present, US EPA recent releases just within the last, well, you can see here, June of 2015. This comes out of the US EPA finally sitting down and putting their thoughts together in the form of health advisories for 2 cyanobacterial toxins, and of course, what do you think those might be? Microsystems and Celinda Opsin. If they'd asked me, I would have advocated for Anatoxin, but I don't think it really matters at this point. Microsystem, primarily. Excuse me. Recommendation for public water systems to manage them, June 2015. Health effects support documents for the individual microsystems, and they have these out for microsystems, Celinda Opsin. They also have them out for Anatoxin and Saxitoxin, and they're all available at this website, EPA.gov. Drinking standards, drinking water standards is where it's all located. Okay, and as I mentioned, where did that impetus come from? Well, I have to give them credit. They were working on this before Lake Erie last summer, but as again, as in a good example, it either takes 130 people to die or maybe 500,000 people to have their drinking water turned off that kind of drives what happens next. And if you haven't heard about Lake Erie drinking water last year, right? A little bit anyway. Well, my professional appointment for 30 years was in Ohio, and I actually didn't ever pay attention to Lake Erie until I was there 25 years because they really never had any issues. I was elsewhere, usually out of country, because out of country they were more concerned about HABs than they were in the U.S. But beginning around 2000, 2001, the blooms started to come back in. We started to pay more attention to them, and eventually we were at the situation. But this kind of summarizes it nicely. By the mid-'80s, Lake Erie point source phosphorus loadings were reduced by more than half from the 70s, and many of the problems associated with eutrophication were reduced and eliminated, and that's true. Just goes to show you, you can do something. But by the mid-'90s, nutrient enrichment led to blooms and has since worsened, and in 2001 we had heavy spring rains flushing a large amount of non-point source phosphorus into Lake Erie. This is right out of the Lake Erie website explanation. This was soon followed by warm temperatures creating a massive algae extending about 5,000 kilometers squared. So here's a 2011 satellite. And of course, this is the western basin of Lake Erie, which is where everything happens anyway, because that's where most people live. And it's the more shallow part. So, if you do impact the system with those factors inducing blooms, then sure, in Lake Erie, this is where it's going to be mostly. But still, it's a big area. And that was Microcystis again. Well, in 2012 then, the International Joint Commission, which of course was established after these events in the 80s, that's what was set up to monitor and deal with all of this, was a joint Canadian-US Joint Commission, established what's called the Lake Area Ecosystem Priority, or LEAP, probably the equivalent of SWAMP, in response to lake-wide changes in Lake Erie related to problems of phosphorus enrichment. And that's what's kind of driven the situation since then, is this LEAP program. As I mentioned in 2003 in a presentation for the Lake Erie Millennium Network, I pointed out the reoccurrence of the blooms. They were coming into Lake Erie at that time. Well, then last summer, 2014, in August, love these political cartoons in the newspapers. This popped up in the newspaper in Cleveland. No, sorry, Columbus Dispatch, not Cleveland. Lake Erie drinking water event in August 2014. And I took this directly, clipped it directly out of the EPA website from one of those documents talking about their recent publications in June, which says in early August 2014, it actually was the weekend, first weekend in August, microcystin concentration at Toledo intake rapidly increased to 14 micrograms per liter. Keep in mind, everybody who does this usually has 1 microgram per liter as the action level, is the acceptable level in drinking water. Well, the microcystin concentration in the finished water, at least what they were willing to report, went as high as 2.5. And it literally did this overnight. They claimed they had no idea the bloom was out there, that it was setting over the intake. The intake for Toledo is about a mile offshore. One of the things you keep in mind is, maybe out here you're newer, back there these water treatment plants were built right around the Second World War, and there hasn't been a whole lot done to them since then. So that's always an issue, because responding and gearing up, doing something besides normal flocculation and alum treatment maybe, and some, upping your chlorine. They don't have advanced ozonation, they don't have dissolved air flotation, they don't have advanced oxidation, or they usually have the ability to do carbon, dissolved granular carbon or powdered carbon, but they only know when to apply it when they need it. And of course, if you don't know you need it, you don't do it. So this is what happened. The microcystin levels spiked, and they were forced to issue an alert and shut down the water supply. Well, since then, they've done quite a bit. They responded fairly quickly and added carbon and did other things. And it was literally 3 days later they were able to reopen the water supply. But in interim, they had to get the National Guard to truck in bottled water, and this was a hot time of the year, and the usual sorts of things. And it's not just drinking water. You think about what else uses drinking water or potable water from a water supply, municipal water supply. You then had all these unintended consequences like, well, this brewery just made 100,000 gallons of beer with this water, can we sell it? This company just milled 100 tons of wheat into flour using water, can we sell it? Things like that. And of course, they're still dealing with that, which then forces you to have to be able to identify toxins in different kinds of matrices. Maybe matrices we never thought about before, like beer and flour. Anyway, so this is what they've dealt with, and this is what's driving the situation since then. Well, in 1980, the US EPA took some of their initial interest in cyanobacteria and funded me to put on an international conference that we held at Wright State, where I was located at that time. And I brought together people from around the world that I had known at that point anyway, and we turned out a book, and at that time as well, entitled Water Environment, Algal Toxins and Health, and that's no longer available, but it did service quite a bit of useful sort of information at that time. We didn't really ever have a fixed international set of symposia for cyanobacteria, but 13 years later, the Europeans decided they wanted to reestablish this. So we had another one in 1993 in the UK, another one in Denmark in 95, and then roughly every 3 years since then we've tried to have another one. So USA, Australia, Norway, Brazil, Turkey, South Africa. So if you want to go to the next one, and I know you all have state budgets that allow you to travel to China, not, but 2016 in September is the next one, next year. And it'll be particularly good because, as you will learn, if you don't already know, China has become the poster country for cyanobacteria blooms. They have them everywhere, in everything. And there are hundreds of people now working actively in China on cyanobacteria, all aspects, management, treatment, testing, engineering, how do you engineer a lake and things like that. So if you, and it'll be in Wuhan, which is in central China, which is where I worked for several years off and on. We had our China project. It's the Hydrobiology Institute. It's not like it was in the 80s. They have very nice facilities now. Okay, there's the introduction. Questions that I can, a lot I'm going to come back to in one form or another, especially taxonomy. Yes?
You mentioned in your cartoon the iron also might be a limiting factor, but it has never been, right?
Some people say it is, but no. I think it's all really coming back. I mean, iron does induce blooms. I mean, there are all these things that, you know, iron is important for blooms. But in terms of cyanobacteria blooms, the controlling factors are really nitrogen and phosphorus. Nitrogen, phosphorus, temperature, stratification. Yeah. Yes?
I understand that most of these toxins are happen when you consume them. What about when there's just skin contact? And then what is the shelf life of the toxin? Can it go on? Does it change in time?
Good question. Yeah. I have a whole lecture on that, but it's not for this workshop. But in general, the microsystems are extremely resistant. They resist boiling. They are sensitive to bacterial degradation in the environment. They are sensitive to photo degradation. So if blooms come up on the shore and scum, and the light hits them, they'll inactivate it. But you only have to go, if the scum is thick, you know, which it often is, you go down in here and it's still active and present. So I guess the main thing is toxins tend to be there all the time. At Santa Cruz, somebody brought a sample in from a lake that they had been treating to kill the bloom. I forget where it was now, but the bloom's gone, but the toxins are still there, and they're wondering, why do we still have toxins when the bloom's gone? And it's microsystem.
So what would be a typical duration of a toxin during a bloom in a lake? If you could follow the toxin, how long do you figure it's going to persist?
Well, if your cell counts are exceeding your, you know, whatever guidelines you have, let's say 100,000 cells per mil, which is often used, and those counts to stay there, the bloom and the toxin will likely stay there, regardless of how long it takes.
I mean,
if the bloom is gone, then it's going to depend on these other things. Do you have organisms that have been developed for? If you're having continuous blooms, if you have year after year blooms, the bacterial population kind of adapts, and you'll get bacteria that like to degrade that toxin. But if these are new blooms, or they don't happen very often, then often you don't have that flora, microflora, to degrade, or whatever other circumstances there are. So the answer would vary a lot, but it could be a few days to a few weeks to a few months, a couple months. In Oregon, most of the time, for example, once the bloom is gone, they don't monitor anymore, so they don't know. And I can understand that if the bloom is gone, your chances that the levels are going to be fairly low anyway, even if they're measurable, may not require it, and then your budget determines what you do or don't do. So, sorry.
How potent are the toxins?
The top potency of the toxins on a weight basis, let's say a microgram of pure toxin per kilogram of body weight, places them in this EPA category called super toxins. In other words, they are lethal at micrograms per kilogram. And a bloom like this could easily be producing several hundred micrograms, maybe even a milligram per gram of cells. And in a bloom like this, a gram of cells might be 100 mils. That's a lot of toxin. That's an extreme situation. But tens of thousands of micrograms per gram have been reported. Normally, most often you're down in the hundreds. I always kind of rule of thumb, once things start hitting about 100 micrograms per gram of dry weight of cells, you have the potential for animal toxicities that would be ingesting the water, somewhere in that range. And of course, the guidelines for drinking water are 1. So that's kind of a rule of thumb. That's in another lecture too. Anyway, I wanted to tell you the iron. Iron is an issue in marine. The reason it was up there is iron is up there for marine HABs, because iron deposition in the oceans is a key driver for HABs in the ocean, not so much in freshwater. Sorry, go ahead.
Could you repeat that again, what you were just saying about the 100?
Well, my rule of thumb over all these, time is once we start seeing levels of toxin of about 100 in the bloom, 100 micrograms per liter or per gram of cells, however you're measuring it, you are getting close to being able to poison animals that would normally drink that water, okay? Whatever that water supply was, you would see toxic effects. You may not see lethality, but with microcystis, you would see liver dysfunction and some of the symptoms associated with it. Yes?
If it has been changing from the non-nitrogen fixers to nitrogen fixers, then how come Microcystis is still the dominant one?
Microcystis is a non-nitrogen fixer.
Right. But I remember in your first, maybe the third slide, you mentioned that it has been changing from non-fixers to fixers, the dominant ones.
Oh, sorry, no, the other way around, from fixers to non-fixers. Did I misquote? Oh, okay. Yeah. Things like Anabaena, Phanozomenon, those are all fixers. They have the heterocysts. So in nutrient regimes where the nitrogen is limiting and maybe phosphorus is not so much, and the nitrogen fixers are there, but once that nutrient level goes up and phosphorus no longer becomes limiting, nitrogen doesn't become limiting, then you shift to the non-nitrogen fixers like Microcystis. And also others too, like Planktothrix, which is another potent producer. You have that quite a bit here in California too. And those both also seem to tolerate warmer waters and longer durations, and Planktothrix survives in ice over the winter, and so they become more of an issue. Yeah. Sorry. Yeah.
Were you involved in any bioaccumulation studies with Lake Erie, or do you know of any that are ongoing right now?
Bioaccumulation with Microsystem, the very first ones I ever became familiar with were actually in Scandinavia, where they were documenting freshwater shellfish, clams, being eaten by wild animals like muskrat in particular, and being poisoned and dying from liver. Really hasn't been a whole lot of bioaccumulation studies done on freshwater at all. Part of the reason for that is the mechanism of action for Microsystem. Generally, the primary consumer of that dies before it has a chance to pass it through. The bioaccumulation, especially with the marine, like saxitoxins, Shellfish are passive. They're immune to saxitoxins, so they just pack it away until something else eats it. So they can build up high levels. Same thing with, like anchovies and mackerel and stuff that are getting into sea lions and things like that here. Now, the other bioaccumulation study, which really wasn't a bioaccumulation study, but had to have been that way, was Pinto and your sea otters, because the washout filter feeders, sea otters. So clearly that was a bioaccumulation, but it wasn't studied as a bioaccumulation study. Yeah. So it can happen, yeah. But it's really, there isn't a lot of studies that are done, like looking at zooplankton